You're listening to Conferences Online Allergy at Children's Mercy Hospital in Kansas City, Missouri. Today is April 8th, 2024. Our topic is Abbas Chapter 12, B-cell activation and antibody production, presented by Dr. Nikita Raje. She is an associate professor of internal medicine and pediatrics and is the chief in the section of allergy immunology at Children's Mercy Hospital.
So again, chapter 12, today is April 8, 2024. That's my email if you need to ask any questions. Disclosures, nothing relevant. We went over learning objectives. Question for today: a previously healthy young European woman who had not been immunized against measles became infected with the virus while traveling in Africa. She developed a rash, fever, cough, runny nose, then fully recovered after 4 days. Blood tests performed 2 days later after her return to Europe showed the presence of high-affinity Ig antibodies specific for measles virus. Which of the following protein was not necessary for production of these high-affinity IgG antibodies? Which of the following events did not occur in germinal centers that formed in the measles-infected woman described in question 1? And which of the following membrane proteins on a B cell generate inhibitory signals that block proliferation and differentiation of the B cell? So we'll get those answers while we talk today. So let's start with the overview of humoral immune response. So when the antigen comes in contact with B cell, it binds to the surface IgM and surface IgD on B cells. Once that happens and B cell gets activated, it's going to proliferate and expand. There is clonal expansion to give rise to maybe more than 5,000 antibody secreting cells, right? And each of these antibody secreting cells can secrete about 2,000 antibodies per second, so that it can keep pace with multiplying organisms that have that antigen. Once they proliferate, either they become plasma cells that will secrete antibodies, they can undergo different, or they can become a memory B cell, and they undergo various processes like isotype switching and affinity maturation. The B cells can secrete 4 different types of antibodies depending on the type of antigen, whether there is T cell involvement or not, whether there was prior antigen exposure or not, and the site where the infection is. So the T cell involvement can be different, it can be varied. There can be T cell dependence for B cell response, or it can be a T cell independent B cell response. It depends on the nature of the antigen too. If it's a protein antigen, it's likely going to be a T cell dependent response. If it's a polysaccharide or other kind of antigen, then it might be a T cell independent response, because remember, T cells can only help with peptides and protein antigens. When there is a T cell dependent response, so in case of a protein or peptide antigen, there is class switching and affinity maturation because of the help from T cells. If it's a T cell independent antigen, like a polysaccharide, these multivalent antigens have repeating determinants, which means epitopes, right? The epitopes are repeating on a polysaccharide. So because of that repeating determinants or epitopes, there is cross-linking of the B cells and they can produce IgM. These are low affinity antibodies, but most of the antibodies produced are the IgM type rather than class switched or high affinity antibodies, as you see in T cell dependent response. So we know that if it's a primary response, which means the first time naive B cells come in contact with an antigen, it gives rise to a primary response, which means it's going to take its time to get activated, and then over time will produce antibodies or long-lived plasma cells or memory B cells. When there is a secondary response, rather than the naive B cells, it might be the memory B cells that get reactivated because of secondary or subsequent exposure to antigen. Because of that, the activation takes shorter time, and this response develops more rapidly, more antibodies that are produced, and they might have the B cells, memory B cells may have the memory of the higher affinity, and they may have already switched to another isotype, so they could produce IgG or E depending on what that memory is, instead of starting with IgM initially. So that's the difference between primary and secondary B cell response. What are the different types of B cells? This is just kind of a um, um, revision of what we have already looked at in the past. The follicular B cells, marginal zone B cells, and B1 B cells. Follicular B cells are the typical B cells that we talk about. They respond very well to protein antigens. Of course, they respond to other antigens as well, but their protein antigen response is very prominent and uses T cell help. So T cell dependence, which means they undergo high affinity maturation and isotype switching. Compared to that, the marginal zone B cells that are found in, say, spleen, mostly they produce T cell independent response to antigens such as polysaccharides or lipids, things like that. B1 B cells are also cells in mucosal tissues, peritoneal cavity, and they also have T cell independent response, which means that most of that response is IgM type and have short-lived plasma cells as a result, compared to follicular B cells that have long-lived plasma cells. Now, this is not absolute, so follicular B cells can have T cell independent response, and the marginal zone B cells can sometimes produce T cell dependent response, but this is for majority of the times. Let's talk about antigen capture and delivery. How do B cells come in contact with the antigen? So remember, antigen, it's present in intact native conformation. There is no processing. There are no antigen presenting cells like for T cells. So that antigen most of the times will be picked up by lymph nodes, by afferent lymphatics, and brought to the lymph nodes by those afferent lymphatics. And over time, those small antigens that are brought by the afferent lymphatics will reach the B cell zone. However, there are some antigens that might be in the blood, go through spleen, and they are bound to complement, and then that way they reach the lymph nodes or lymphatic tissues. Macrophages that are resident in the lymph nodes can also take up larger antigens, but all of these antigens are not really processed for B cells as they are processed for T cells. So how do these, so the antigen reaches the B cells. It eventually reaches the B cell zone where that antigen is taken up by the follicular dendritic cells in the B cell zone, so that it can, so FDCs, so that it can express it on its surface for presentation to the B cells. So follicular dendritic cells are nothing similar or not related to the traditional dendritic cells that present antigens to the T cells. These are follicular dendritic cells are present in the B cell region, B cell follicles, where they produce CXCL13. B cells, when they are moving from one lymphatic tissue to another, remember B lymphocytes are lymphocytes, so they undergo recirculation. Naive B cells recirculate from one lymph node to another, looking for the antigen it can bind to. When they enter the lymph node where the follicular dendritic cells are expressing the antigen on their surface, they might be able to attach to that antigen. Naive B cells express CXCR5 on them, so they move from one lymph node to another and get attracted to the CXCL13 in the B cell zone or B cell follicles in the lymph nodes. That's how they end up in the follicles rather than anywhere else in the lymph node. So we talked about macrophages taking up large microbes and antigen antibody complexes, right? Macrophages in the subcapsular sinus here, shown here, that will take up larger antigens, whereas the smaller antigens will be brought in from the afferent lymphatics. They go through the various sinus, lymphatics, afferent lymphatics going through the subcapsular sinus and finally reaching the B cell zone. All right, and then the complement receptors also are present on the follicular dendritic cells or marginal zone B cells, so that those receptors can bind to immune complexes that have complement fragment and the antigen that is tagged by that fragment. So they can actually pluck those, and so because of that, the follicular dendritic cells are taking up that whole antigen bound to the complement fragment using its complement receptor. And that's how then they are going to use that antigen to present to the B cells.
So Dr. Raje, do the naive B cells, do they make the CXCR5 and the CXCR5 guides them to the follicle?
That is correct. It's just not exclusive to naive B cells. CXCR5 is like, so any kind of chemokine receptor is like putting it on your GPS. If it shows CXCR5, it's going to take them to B cell zone. It doesn't matter whether it's a naive B cell or it's a T cell, it's a dendritic cell, whichever cell is going to express CXCR5 is going to be attracted to the B cell follicle because that B cell follicle has CXCL13. Okay. So for now, and we'll come to that in this same chapter, but for now we are talking about naive B cells. They express CXCR5 on them, so they will go to the B cell zone.
Yeah, thanks.
Okay, and when we talk about CXCR5, we are typically, if we are talking about B cell zone, right? And so we are typically talking about B cells, but you'll see a variation why I'm saying it's not exclusive to B cells. All right. So the antigen has entered the lymph node. B cells, naive B cells, are recirculating from one lymph node to another. And if it comes across the antigen that it's specific for, which means cognate antigen, if it comes across that cognate antigen, it's going to be able to bind to that antigen and the activation process begins. So that's the first signal. BCR binding, what's BCR? It's surface IgM and IgD, right? So BCR binds to the antigen using one particular epitope. Now, that first signal may not be enough for that BCR to be activated. And so it has to have that second signal that's typically provided to it by innate immunity, right? Who provides the second signal in T cell, B cell activation? It's the innate immune system. So in case of T cells, what's the second signal? Rather than second signal, what binds to, what's the co-receptor? Let's talk about that.
The co-receptor for T cell, like in T cell activation?
For T cells, yeah. What are the co-receptors? Very easy. What are the co-receptors? Not CD3, but CD45? No, co-receptors. So we are talking about.
Oh, you mean like the B71 B's, like the.
That's second signal. So B71, B72 bind to CD28. That's your second signal. Before that, the CD3 binds to, or the TCR binds to the antigen. And then the CD4 or CD8 also binds to the MHC part, right? So the co-receptor also interacts with the same antigen that the TCR binds to, which is either CD4 or CD8 for T cells.
Okay.
Right? CD4 or CD8. Those are called the co-receptors. So here, the co-receptor for B cells is CR2, CD19, and TAPA. This is the co-receptor. I hope you guys can see my pointer here. So the antigen binds to the BCR using a particular epitope, and then it has a complement fragment C3d bound to it because it was opsonized by this fragment. Complement has opsonized this fragment, and the co-receptor includes complement receptor CR2 or CD21. So that complement receptor can bind to the complement fragment that's bound to the antigen. So that That way, the co-receptor binds to the same antigen that the receptor binds to. Same way, in T cells, the TCR binds to the antigen, and the CD4 or CD8, depending on the different types of T cells, will bind to the same complex of the antigen that has the MHC complex, right? So MHC binds to CD4 or CD8, MHC class 1, class 2, remember those things? So they bind to it. Similarly, BCR binds to the antigen, and the co-receptor of BCR, which is the whole complex here, will bind to the complement fragment using the CR2. CR2 is part of the is the part of the co-receptor for B cell. That makes this whole complex or whole interaction pretty stable. Was there a question?
Yeah, the C3D, the complement is just floating around in all the lymph nodes.
Not in the lymph nodes. So it's a complement. So it's in the circulation. So say in the spleen, the it's filtering the blood, and the blood has the antigen. If the blood antigen is in the blood, it's gonna be tagged by complement. Right? That's how complement works. And so a fragment that tags or opsonizes the antigen because of complement activation, one of them is C3D.
But this would still, it gets tagged in the blood, but then it would still go to the lymph node, right? Or an lymph
Right,
organ.
lymphatic tissue. It can go through spleen, then end up in the lymphatic tissue, and eventually it pretty much circulates to lymph nodes.
Got it. Okay.
All right. So here is the receptor binding to the antigen, co-receptor binding to the antigen. And just like T cells, once there is interaction between TCR, the co-receptor CD4, CD8 with that antigen and MHC complex, then the next part is second signal. So again, here, the signal is provided by innate immunity such as TLR. So PAMPs, or like the antigen, has pathogen-associated molecular patterns that bind TLRs, and that TLR activation or signaling will kind of help the T cell to make sure that it's only reacting to an antigen that is a danger, right? That's the purpose of the innate immunity, providing that second signal that the T cell activation is not being done in vain. So that helps giving that second signal that the activation is needed. Once that activation happens, so once that also there can be other signals for B cells, like TLR secrete APRIL or BAFF. These are B cell activating factors. BAFF is B cell activating factor. APRIL stands for a proliferation inducing ligand. So both of them help with the B cell activation. Once the B cell is activated, what happens? What are the changes in this naive B cell? Naive B cell interacts with the antigen, co-receptor interaction, TLR signaling, all of that happens. Once that happens, there is proliferation, and then there is differentiation. During that process of activation, there is increased expression, there is change in the expression of some molecules. The first one is increased expression of CCR7 that helps in it to migrate to T cell zone. So remember, CXCR5 was on naive B cell. Now it starts expressing, instead of CXCR5, it will start expressing CCR7. CCR7 expressing cell is going to go towards the T cell zone where there is CCL19. So it starts moving towards T cell zone so that it can get help from T cells. It increases the expression of B7-1, B7-2 so that it can interact with T cells with that interaction between CD28 and B7-1, B7-2. It will increase expression of receptors for T cell cytokines so that the cytokines released from T cells, say helper T cell Th2 is helping this cell. It's going to express cytokines like IL-4, IL-5, IL-13, and so that will help in class switching these B cells, right, to say IgE or IgA, depending on what the antigen is. So overall, that TH help is received in form of cytokines, and so when the B cells get activated, they increase expression of cytokine receptors. And there is increased expression of anti-apoptotic proteins so that there is increased survival of the B cells. So that's the change once the B cell gets activated. Now, depending on the type of antigen, there might be help from T cells or may not be there might not be help from T cells. If it's polysaccharides or lipids, it's T cell independent response, and they will have multiple identical epitopes that will cross-link the BCRs on that B cell and help in initiating proliferation and differentiation and production of antibodies, which are mostly IgM type. T cell dependent process, it's a protein antigen. It possesses only one copy of each epitope, so it is not able to cross-link the BCRs, but there is help from the T cells. So those T cells can actually be helped in two ways. So it's like a bidirectional help where the B cells express those antigens on their surface and present them to the T cells, and T cells in return help with affinity maturation and class switching. So here's our T cell dependent response. There is recognition of antigen. So there are naive T cells and naive B cells in the lymphatic tissue recirculating from one lymph node to another. Dendritic cells, if there is an infection, that antigen is picked up by dendritic cells, and they bring that antigen to the T cell zone, where they present that antigen to the helper T cells. Also, the naive B cells get activated using some other epitope of that antigen and activate the B cells. Those T cells are going to move towards some of those T cells are going to move towards B cell zone because they'll now express CXCR5, and the B cells are going to start expressing CCR7, so they are going to start moving to T cell zone. So both of them interact at that at the periphery of that T cell zone and B cell zone, and that interaction helps in class switching, production of short-lived plasma cells, and production of follicular helper T cells. Those follicular helper T cells are going to help in the process of germinal center reaction, so affinity maturation and memory B cell production. So we talked about this, I just mentioned it. What's the role of chemokines? There are several naive B cells and T cells recognizing the same epitope. That could be 1 in 100,000 or 1 in million lymphocytes that's recognizing the same antigen. So what happens is the follicular dendritic cells in the B cell zone, B cell follicles secrete CXCL13 that attracts any cell that expresses CXCR5. When the T cell and B cell get activated, the T cells, helper T cells that are going to help B cells, they are going to reduce their CCR7 on their surface and increase CXCR5, so they move towards B cell zone. B cells, on the other hand, on activation, reduce the CXCR5, increase CCR7, move towards T cell zone that expresses CCL19 and CCL21. So at the periphery of that T cell zone and B cell zone is where they interact. All right, when the B cell comes in contact with the T cells, it becomes, remember, activated B cells become antigen presenting cells. So these activated B cells now take up that protein antigen and process it. They process it and then they express the peptides bound to class 2 MHC on their surface, and they present that antigen to the helper T cell. When they present that antigen, they will have response. They might have interaction using B71, B72 with CD28, CD40 interacts with CD40 ligand on the T cells, and all of that interaction helps in the communication that activates the T cells. So B cells activate T cells by expressing or presenting that antigen, and T cells in return, using the CD40 ligand interaction, will help the B cells in their function. So that CD40 ligand, CD40 expression. So CD40 is on activated B cells. CD40 ligand, on the other hand, is expressed by activated T cells. Sorry, I take that back. So B cells always express CD40, and T cells express CD40 ligand only on activation, right? So even though B cells will have CD40, they won't get help from T cells unless the T cells are activated. And when that interaction occurs, there is activation of transcription factors, there's translocation of transcription factors like NF-kappa B and AP1 to the nucleus. And what are those transcription factors? Those are inflammatory transcription factors that lead to activation of NF-kappa B pathway and AP1 and lead to inflammation. So mutations or variants in CD40 or CD40 ligand lead to X-linked hyper-IgM syndrome with defects in class switching, because of which there is high IgM but low G, A, or E. All right, so remember, apart from, so most of that antibody response comes in from the follicles where there is a germinal center that's created in those follicles, and there is T cell help. But there is also extrafollicular antibody response. So initially during the B cell activation, the B cells do get activated and start producing antibodies outside of their follicle, which is extrafollicular antibody response. There's limited class switching and short-lived plasma cells that are produced. About 4 to 7 days into that interaction or activation of B cells, there is germinal center that's created in the primary follicle. Once the germinal center is created, it's called a secondary B cell follicle. During that time of germinal center production, there is that interaction between follicular helper T cells and B cells that help in affinity maturation and isotype switching. There's production of long-lived plasma cells and memory cells. That's the purpose of the germinal center response. So let's look at what is germinal center response. All right, so there is formation of sorry, I'm looking at the time here, formation dependent on CD40 ligand CD40 interaction, which creates that germinal center. And there are various zones of the germinal center. There is a dark zone and a light zone. So the dark zone in the follicle. So this is a B cell region, B cell follicle, where there is formation of germinal center. In that follicle, there is a peripheral dark zone where the B cells are rapidly proliferating. These are called centroblasts. And they proliferate so quickly, their doubling time is 6 to 12 hours. And the cells that are produced, the centroblasts, are later moving towards the light zone where they're called centrocytes. So centroblasts in the dark zone, centrocytes in the light zone. In the light zone, so where these cells are proliferating, they move to the light zone where they undergo affinity maturation. And how does that happen? The follicular dendritic cells are sitting in the light zone and they are presenting antigen. And with the help of the hypermutation that's occurring in that dark zone, the B cells with different affinities come to the light zone where they get tested whether they can bind with high affinity to that antigen or not. The ones that bind with higher affinity survive, and the B cells that don't have higher affinity kind of start getting deleted, so that there is at some point only the higher affinity B cells are going to survive. So that's called affinity maturation. All right, so, and then there is mental zone where there are naive B cells. So the naive B cells are sitting there waiting for the antigen. When it gets activated, it makes that germinal center, moves to a place where it continues to proliferate. That creates the dark zone, and then it gets tested against the antigen presented by follicular dendritic cell in the light zone. So that's your germinal center. Activated B cells migrate into the follicle, they proliferate actively to form that dark zone, and they undergo class switching and somatic hypermutation. Then they migrate to light zone, and get tested against that follicular dendritic cell. And the ones that survive and have higher affinity will move on to become a memory B cell or plasma cell and exit the follicle. All right, we talked a little bit about this, how the follicular T cells are generated. This is important. This requires initial activation of dendritic cells, right? T cells get activated because of presentation of antigen by dendritic cells. And depending on the transcription factors and various signals like cytokines or other receptors, there can be creation of follicular T helper cells. Once that initial activation occurs, the subsequent activation is by activated B cells. We talked about this. Activated B cells become antigen presenting cells for T cells, so they present that antigen to T cells, and there is interaction between second signal. Now, instead of just CD28 and B7 and B72, the other interaction is between ICOS and ICOS ligand. So the ICOS ligand on the B cells binds to ICOS on T cells, and that helps in the follicular helper T cell stabilization and its function. And there is another molecule called SAP. We talked about this. SAP stabilizes the expression of transcription factor BCL6, which helps in function and proliferation of T helper follicular T cells. Defect in SAP, we know, causes XLP type 1. The cytokine that's important for these cells is IL-21. That's important for generation of germinal center and creation of plasma cells. So what's isotype switching?
For XLP, Dr. Raje, so the consequence would be that we would have less class switching and less IgA, IgG, IgE. Is that right?
So it's not like hyper IgM, right?
Okay.
But it causes dysregulation that leads to proliferation. There is lymphoproliferation because of that.
Okay.
All right. So for isotype switching, it occurs in the extracellular foci driven by local T helper cells, but most of it occurs in the germinal centers. And so switching is regulated by cytokines made by helper T cells, and each of the isotypes have particular effector function. So, but the specificity of the antigen that they can bind to is still the same. So what is the mechanism of class switching? The CD40 ligand interaction leads to activation of AID. AID, activation-induced deaminase, is important in this class switching. There are cytokines and switch regions that take part in it. So switch region, so VDJ recombination, we read about that. That happens during the development of B cells. Class switching occurs after the B cells are already mature and they are activated by an antigen. At that time, class switching occurs, and there is a switch region before each of the constant region. So S mu, S gamma, S epsilon, S delta, or S alpha, all of these switch regions are at a locus that's 5' to its constant region. And there is also an I exon and I region promoter that's also 5' to every switch region. So here is the S region, S mu, and there is that I exon that's and a region, I region promoter that's 5' of the switch region. And so the aid is important in switching so that the region between the VDJ is bound to the C mu region. So the initial antibody is IgM, but when it needs to class switch, it will cut out the portion from one switch region to the other switch region where the other constant region is located. That could be C gamma or C epsilon, depending on whether we are trying to make IgG or IgE or IgA. So the rest of the DNA in between those is going to be cut out, and then the VDJ region joins this constant region of the new isotype. So how does that, so this shows the same thing, S mu region, I mu region. For each of these constant regions, there is switch region and the I exon there, and the part of the DNA is cut out when the class switching occurs. So what happens at the site of the break? So there is RNA DNA hybrid formation in the switch region, and in that place, AID deaminates the cytosine molecule or nucleotides at each of these sites. So all of these C's are turned into U's. And then the UNG, uracil N glycosylase, comes along and takes away the U's to make A basic sites, which means there is no base or no nucleotide at that region. Then comes along APE1 nuclease that will make NICs at those places, so the breaks are created. And then there is rejoining so that the final DNA is created and the switch region is taken out. That was the class switching. The second type of process is affinity maturation. So class switching, what does that do? The one that we already talked about, class switching changes the isotype from IgM to G, A, or E, depending on the antigen that they came across, right? That class switching occurs in the germinal center with the help of follicular helper T cells. So now we talk about the second process that occurs in the germinal centers, which is affinity maturation. The isotype switching occurred at the FC, the constant region. Now, affinity maturation, on the other hand, happens in the FAB region of the antibody, right? It's the antigen binding site that needs to change so that there is increased affinity of antibodies for a specific antigen. This occurs because of somatic mutation, which means the mutations are not occurring in the germinal germline genes, but occurring in the immunoglobulin genes, where there is so many mutations occurring in the V genes of these antibodies that it changes the affinity over time. This again requires help from T cells and that interaction between B and T cells using CD40 ligand. Mutations are occurring at 1 in 1,000 in the V region genes, so it's higher than mutation that you see in other genes and other cells. So this is called somatic hypermutation because that mutation is way faster than any other cells or any other genes. This can result in change in up to 10 amino acids that can be changed, and that can significantly change the affinity with which these antibodies can bind to those antigens. Now, again, this occurs in the dark zone of germinal center, and then they move, once they have changed and there is mutation, they move towards the light zone of the germinal center, where they're going to be tested against the antigen presented by follicular dendritic cells. And the higher affinity B cells are going to survive more, so that over time the affinity continues to mature. So the somatic mutations occur more in the CD3 regions of the heavy chain variable region and light chain variable region. Mutations occur primarily in antigen bindings like the CDR regions, more in IgG than IgM. There is presence of mutations that correlate with increasing affinity, and we saw that the same genes that are, or same enzymes that are instrumental in class switching are also important here. So AID plays a role, and here the U's convert to T's during DNA replication, and they kind of generate the SNPs, or so there is polymorphism at that place. So the migration again occurs to the light zone after somatic mutation, where they come across the follicular dendritic cells and interact, and the B cells that have higher affinity escape apoptosis. So the figure shows how the one that actually is able to survive continues, and then the rest of the cells may undergo apoptosis. There are two types of plasma cells that can be created: short-lived and long-lived. Short-lived can have T-independent or early T-dependent response, and more extrafollicular response that these provide, versus long-lived have T-cell dependent response. These are the B cells that plasma cells that develop and function in the bone marrow and produce large amount of antibodies when they are reactivated. This is showing the structure of plasma cells that actually has a lot of variation from a B cell. The cell becomes larger and there is higher, the ratio of cytoplasm to nucleus changes. Endoplasmic reticulum become more prominent. There is increased immunoglobulin production from these cells, and there's switching from membrane IgG that's seen in B cell. On the surface there is IgM, IgD. Instead, those plasma cells have secreted IgGs. All right, so these immunoglobulin molecules, they can be different types, membrane IgM or secreted IgM. Membrane IgM has that piece that goes through the membrane. So that's the difference. There is difference in that cytoplasmic tail, what it looks like. There are various, these lysine and valine and amino acids that take part in formation of that membrane IgM piece. Versus, and so that's based on the change in the mRNA, and that is that transition because of the alternative RNA processing, because of which that either there is membrane IgM created versus secreted IgM created. So the mRNA undergoes changes. Memory B cells are generated mainly in germinal centers. They express higher anti-apoptotic proteins like BCL2. They also express high affinity BCR and immunoglobulin, and are capable of eliciting rapid response to subsequent exposure to antigens. There is effective vaccines that target memory B cells, require T cell help. So example would be conjugate vaccines. So they have a polysaccharide chain, but it's linked to a protein carrier so that it can give a long-lasting memory B cell response. B cell fate, how do you know when it's going to be short-lived or long-lived plasma cells or memory B cells? It all depends on the transcription factors. BCL6 gives rise to a creation of plasma cells, right? There is transcriptional repressor, maintains germinal center reaction, and prevents cell cycle arrest and apoptosis. BLIMP1 and IRF4 will help in committing to plasma cell fate. So the memory B cell is BCL6, and BLIMP1 and IRF4 are give rise to plasma cells. We talked about the T cell independent response. I'm not going to look at that again. We know that marginal zone B cells and B1 B cells give rise to T cell independent response. Nothing new here. And natural antibodies are present against anti-ABO blood group. So these are the ones that we check. Isohemagglutinins if we cannot check vaccine response, if the patient hasn't had vaccines. These are low affinity antibodies for most part. There can be IgG. Some of those have IgG antibodies. Most of them are low affinity antibodies that we are checking for. And then humoral response needs some regulation. It uses feedback from antibodies, soluble IgG, complexes with secreted IgG, complexes with antigens. The complex binds to Fc gamma R2b or CD32. Remember this one. This is the inhibitory receptor for B cells. It activates signaling cascade that inhibits B cell activation. So because it has ITIM on its surface instead of ITAM. All right, that was the last one. We'll go through questions quickly here. European woman went to Africa, got measles. What's the process that's protein that is not going to be needed for high affinity IgG antibodies?
CRP.
Okay, perfect. You need CD40 ligand, AID, class 2 MHC, CXCR5. Which of the following events will not occur in this woman? After the measles. What's something that happens before versus after? In the interest of time, I'm going to give you
I
the
would
answer.
say,
Go ahead.
I'd go with D.
It's actually B. VDJ recombination occurs during maturation of B cells, creation, development of B cells, not after activation. It's the somatic hypermutation and B cell proliferation, helper T cells, follicular helper T cells helping with somatic mutation, as well as isotype switching and generation of memory B cells, all occurs after activation by an antigen, in this case measles, but the VDJ recombination has occurred during B cell development. And the last one, which of the following membrane proteins on a B cell generate inhibitory signal in B cells?
Fc gamma
This one should
RGB.
be, yeah, A.
Okay, that sounds good.